So you have learnt about CPD photolyase. These are enzymes which are involved in the photoreactivation phenomena. So let us see the mechanism of this CPD photolyase. CPD photolyase can bind to DNA in the dark by recognizing the altered DNA structure, which alteration is caused by a CPD formation, cyclobutane pyrimidine dimer formation, rather than a specific nucleotide sequence. So these enzymes, they do not identify the sequence of DNA to bind. There is no any specific sequence where this CPD photolyase bind. Rather, these are the altered DNA structure that is due to the formation of CPD. This is the site which will be identified by these enzymes, and these enzymes will bind with this site. This binding is about 10 to 5 times tighter when a DNA segment contains a CPD, when a dimer is present, than when it does not present. So it can bind to DNA which is normal DNA and which doesn't contain dimer, but this binding is several times, 10 to 5 times tighter if this dimer is present. Half of the binding energy appears to come from the interactions between enzyme and the DNA backbone. So some energy is required for binding the enzyme with the DNA backbone. So from where this energy comes? So half of this energy comes from the interaction between the enzyme and the DNA's backbone. The other half of the energy comes from the interaction between the FADH at the active site of the enzyme and the CPD. So the other half of this energy is provided by the interactions of FADH, which is present in the active site of the enzyme. However, the light harvesting antenna, which is chromophore, antenna pigment, chromophore, doesn't influence binding. It has no role in the binding of CPD photolyase with the DNA. Once the enzyme DNA complex is formed, the CPD is flipped out of the DNA double helix and into the active site. So once this enzyme binds to the DNA, this DNA, and here a dimer is present. So after this binding, this dimer flips out from the helix. This is helix of DNA. So it flips out from the helix and it comes out into the active site of CPD photolyase, into the active site, into the pocket of this enzyme. After the CPD flips into the enzyme's active site, the energy of an absorbed photon is transferred from the light harvesting antenna pigment, that is chromophore, to the FADH. So once this dimer CPD, this flips into the active site of enzyme, the energy which is absorbed by the photoantenna pigment, that is transferred to FADH, which is present in the active site. FADH then transfers an electron to the CPD to induce cyclobutane ring cleavage. So look at the sequence. The energy, first it this CPD, this dimer, flips into the DNA, into the enzyme's active site. Then the energy absorbed by the chromophore, that is transferred to FADH, and then FADH transfers electrons to this dimer. When the electrons are transferred to the dimer, the bonds present in the dimer, they cleave. The catalytic cycle is completed when the electron is transferred back from the repaired thymine to the FADH cofactor. So this FADH minus, it transferred electron to a dimer, which is thymine dimer. Okay, so after the breakage or cleavage of the bonds between these dimer, this electron is reversed to FADH. So here you can see this sequence of events. So this is FADH, and this is dimer on the DNA. This is DNA helix. So when they bind together, this dimer flips out of the helix. Okay, so then photopigment transfers energy to FADH, and then FADH transfers electron to this dimer, and here you can see the bonds are being opened. These two bonds, these are being opened, and once these bonds are opened, these two thymines 1 and 2, these thymine are now not the part of dimer, but they are separate thymine bases. So once they have been separated, the cyclobutane ring has been disrupted, the electron is reversed back to FADH, and this completes the sequence. So in this way, our dimer is changed into two thymines.